Hi.
Welcome back. Your DNA's been extracted from your cells.
And today, we get to the headliner of the show,
Genotyping. Genotyping is how we figure out things like your ancestry composition and genetic health risks from your DNA.
How do we do it? We read your DNA like a book.
But we don't actually read every single line of the billions of DNA letters in your genetic code.
Oh. We only need to look at around 600,000 specific spots in your genome, a tiny fraction of all your DNA.
And if you're wondering why we pick those specific variants, it's because they hold particularly interesting information about your ancestry or traits, and they often differ from person to person.
Over the past few decades, scientists have perfected this amazing technology that allows us to read specific spots throughout your genome. It's called genotyping, and it starts with a chip. A genotyping chip.
Unlike a potato chip, this chip is perfectly flat, rectangular, kind of like a stick of gum.
The chip is the key to unlocking your genetic code because it holds a key ingredient, synthetic strings of DNA called probes.
Probes are almost like strips of microscopic Velcro, but instead of being made of tiny plastic loops, these DNA strips are made of A's, G's, C's, and T's, the same letters your genes are made of.
These DNA Velcro strips help us locate the parts of your DNA that are just next to the letters we want to read, called variants.
So when we add your purified, chopped up DNA to your chip,
it's a free-for-all. Millions of tiny chunks of your DNA are swimming around looking for their matching DNA Velcro probes. Kind of like trying to find your tiny genetic soulmate in a huge crowd.
Aw, that's sweet.
But it can take a long time to find your perfect match in a crowd. Fast forward about 24 hours, and by then, most pieces of your DNA have found their Velcro probe soulmates.
Once a piece of your DNA hooks up with its Velcro probe soulmate, we want to look at the letter in your DNA that comes just after the end of the matching probe.
And to do this, we use colorful fluorescent dyes.
Ooh, color!
We wash your chip with a mixture of fluorescent red and green dyes that are attached to loose DNA letters.
So A's and T's are labeled with red, and the C's and G's are green.
And whatever color letter combo sticks to the end of the probe tells us which variant you have at that location.
Well, at this stage, technically it's still just a lot of information about the colors associated with each of the 600,000 plus variants that we care about in your genome. And we decode that color pattern in the next step, analysis.
Yep, and there's one final quality check to make sure that enough of your variants have a color associated with them.
But once it passes this step, we send all that color information to the analysis team, and they'll figure out what all those colors can tell you about your genetics.
Stay tuned.